desktop if you have a desktop in your room. Who owns a desktop nowadays?
A desktop? Oh.
I mean, my family has one like at home.
Debbie's like, a desktop? What is that?
I had to think about it for a minute.
Desktop computer.
Video games.
Man, Gabby, that's pretty judgy.
No,
Desktop.
no, I was like thinking about what that meant.
So,
Oh my goodness.
I don't know if you guys are aware, but there's a biology department Facebook page, and things like internships and graduate student opportunities and stuff like that turn up on that occasionally. Today there was a PhD student who is working at Stowers in their PhD program. One of the mine students that Stowers has a PhD program and pays like $37,500 per year, and they give you a computer. And I commented on the post, when I was a graduate student, they paid $9,600, and you have to pay tuition and fees out of that. And then she writes back, oh, and you don't have to pay tuition or fees. And then I'm like, and we have to buy our own computer. So things have changed since the early 90s.
I can't find it. Is it just called brain fuel biology group or what is it?
Oh, I have no idea what it's called. Just because I set it up doesn't mean that I have any idea what it's called. WJC Biology Department. That is not what I want to talk to you about today, though. Today, what I want to talk to you about is lab next week. For those of you meeting with Dr. Judge tomorrow, you're going to talk about your experimental design. We're going to rip it and shred it and then build it back up. And you'll have lots of changes that you'll need to make. So next week, you need to come in with a completed experimental design based on what you talk about in lab this week. And the other thing that we're going to spend time in next week in lab is talking about statistics, and we have a couple of data, well, we have one data set that we can analyze a couple of ways. We're going to do all of this in R. So you need to bring your computers, if you have laptop computers, to class in lab on Thursday. And we're probably going to meet in a different room. I'll notify my students of where a room is, because it would be good to project things, and we don't have that ability in the lab. And so you need to download R and RStudio by the time you get there, so that and open them up and make sure that they work correctly. So on the Moodle site are a couple of scripts, simple scripts that you can use to visualize the FinchBeak data set. I put that up, the data we went through, principal component analysis, and so those scripts are there and available for you. Open them up, make sure that R works, so that we don't spend an hour and a half going around troubleshooting everybody's computer to make sure that R is functioning on it. If you download R and RStudio and then you open it up and it's not working, come see Dr. Judd or I about getting that up and running. A lot of times it's a simple thing of you're running R in one directory and you're trying to access data and stuff that is in another directory, and you either need to tell R to do that, or you need to put all of your data in the same directory where R is. So a lot of times the fixes are simple, but it's better to get those things worked out beforehand rather than trying to do it. There are instructions in the syllabus on where to go and get R. Base R is at the CRAN website, which is the Comprehensive R Archive Network. If you have a Windows machine, there's a version for Windows. If you have a Mac machine, there's a version for Mac. I doubt that any of you are running your computers in Linux, but there's also a Linux version. Base R is Base R. You can run out of Base R, but I find RStudio a little more intuitive. It's free, do not pay for it. Go here, download RStudio, just click on download RStudio, and it will take you to a download. You want the free version, and I think it actually even reads what kind of computer you're on, so it just gives you the option of downloading for Windows or downloading for Mac. Get those two things up and running. will be ready to go next week in lab. Some of the graphs that I'm going to show you today were actually generated in R. If you would like, I can put those scripts up on the Moodle site as well if you would find that of interest or of use. But there are already some scripts up on Moodle that you can use to test and see if it's working.
I know, Dr. Kowinski, did you put the cat on the PowerPoint just for me?
I did not. I put a cat on the PowerPoint because that is a mixed cat. It is a cat with a genetic condition that makes it this dumpy cat. It's got a short body, squat legs, and no tail. So we'll talk about Manx cats a little bit later in class. So I asked you to do a trihybrid cross because I'm evil. Did you guys do your trihybrid cross?
Yes.
What did you notice about the trihybrid cross?
It's a pain.
It's a pain, it's a lot of squares. I'm thinking about genotypes.
As a ratio.
Hmm?
As forms a ratio.
How do you mean? What do you mean?
So, based on phenotype, you can tell what phenotype it will be,
Yep.
and there's a very distinct amount of variation.
Yeah, so that symmetry that you saw in a dihybrid cross, when you were working with two traits, where you had one upper corner was the homozygous dominant individual, the lower corner was the homozygous recessive individual, and as you went diagonally across the table, you got fewer and fewer dominant alleles in your genotypes. That same type of symmetry that is in a dihybrid cross when you form gametes using the FOIL method occurs in this table as well. So the reason that I asked you to do this is because today we're going to be talking about quantitative traits. And so learning goals for today is we want you to understand what these polygenic traits are and how they undergird a lot of what we think of as relevant phenotypes for evolution, and how this is all based on essentially Mendelian genetics. We're going to review the Hardy-Weinberg equilibrium and equation and how it works, and we're going to go through Hardy-Weinberg equilibrium and show you how you can use Hardy-Weinberg equilibrium equation to model what happens with evolution over time. And then we're also going to teach you the Breeder's equation, which then deals with these quantitative traits and allows you to project what the rate of evolutionary change is in quantitative traits, and we'll also take you through more detail of how you go about estimating what the heritability of traits are. So there's a point in the PowerPoint presentation where I'm going to stop and ask you a question. Don't go to the next slide when we get there, because the next slide has the answer. So the PowerPoints are up on the Moodle site, but don't just go blazing through them, because I want you to actually think about the answer before you give me the answer. So this is what I gave you before. You have two homozygous individuals, homozygous dominant for all loci and homozygous recessive for all loci. What's the first step in doing any genetic cross? Well, yeah, but what do you have to know? If you're going to see how this one and this one combine, what do you have to combine?
One letter to the other one letter to the other one.
Yeah, what would we call those things that have letters? They're haploid, not diploid.
Alleles?
Genotypes?
Not genotypes. These are genotypes. Homozygous dominant, homozygous recessive. We have to form gametes. So the first thing you have to do is form gametes. What gamete types can this individual make? They're all dominant. It'll be a dominant A, dominant B, dominant C. What kind can this individual make? All recessive. Recessive A, recessive B, recessive C. So I always circle my gametes simply because that keeps it straight in my mind. This is a gamete, it's not a genotype. So I'm always inventing strategies for myself to keep myself straight. And then when we take this gamete, combine it with this gamete, we get the genotype of the offspring. And this is where I left you yesterday, kind of. If we take these individuals and cross them, what are we going to get? How should we go about the process of finding out the gametes that this individual should make. How are we gonna make gametes here? Yeah. Did you have your hand up?
Well,
Yeah.
so you just mix them. You can
Okay.
make it if you put it with a big B or a big B.
Okay, so how did we do it for this?
Big A, big B, big A, little B, little A, big B.
Yep. First, outer, inner, last, right? Just like you do. If we want to solve this, how do we do it? We multiply first, then we multiply outer, then we multiply inner, then we multiply last. Same thing in forming gametes. So we would have this, but then we have to do this twice because one half of these gets the big C and the other half gets the little c. Right? So we have 8 gamete types.
I think your PowerPoint has a mutation.
My PowerPoint has a mutation. Oh my god, my PowerPoint does have a mutation. That should be a C, shouldn't it? I never noticed that before. Alright, so what you should have gotten was this. And then you should have gotten this. But as Dr. Judd was telling you at the very end of class, some traits are determined ultimately by a bunch of different genes, each gene contributing a little bit to the phenotype, adding a little bit to the phenotype. So in this case, is it important what the genotype is, or is it important how many dominant alleles you have? What's important is the number of dominant alleles you have. So here we have 6 dominant alleles. Here we have 5 dominant alleles. You'll notice that this genotype is the same as this genotype. In this third diagonal here, we have one genotype here, another genotype here, and a different genotype here. When you compare this genotype to this genotype, it's flipped with the dominant A's, recessive B's versus recessive A's and dominant B's. If you go across the diagonal here, it's 1, 2, 3, 4, 5, 6, 7, 8. 8 things along the main diagonal bisecting this Punnett square. They're all heterozygous for both traits. You do the same thing. You see that all recessives are down here. This diagonal has 2 individuals of the same genotype. This diagonal has 2 individuals of the same genotype and 1 individual of the different genotype. The symmetry in the table is similar to the symmetry that is in the dihybrid cross table. But if all we care about are just the number of alleles that contribute to a trait, do we have to worry about the genotypic ratio of this monstrosity? No. All we have to do is count up the number of alleles, which I have done for you. 6, 5, 5, 4, 5, 4, 4, 3. Oh, guess what? 6, 5, 5, 4, 5, 4, 4, 3. 5, 4, 4, 3, 4, 3, 3, 2. Oh. 5, 4, 4, 3, 4, 3, 3, 2. There is symmetry to the table. But what matters to you is how many individuals in the 64-cell Punnett square have 6 alleles, or 6 dominant alleles. Just one of them, right? How many have 5 dominant alleles? 1, 2, 3, 4, 5, 6. How many have 4? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15. How many have 3? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, wait a minute, I miscounted. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So what were those numbers? Those numbers were 6 alleles has 1, 5 alleles has 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Six alleles was 1, 5 alleles was 1, 2, 3, 4, 5, 6. 4 alleles was 15. 3 alleles was 20. Because there's symmetry in the table, do we have to count up the number of individuals with 2 dominant alleles, 1 dominant allele, and 0 dominant alleles? Why not? It'll be symmetric, so we should expect to find 15 with 2, 6 with 1, and 1 with 0. Is that true? Let's count up the 2's. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15. It works. What does this start to look like? It starts to look like a normal distribution if I've drawn things well. Normal distribution. Whenever you see a normally distributed trait, your mind should first and foremost go to thinking this is a polygenic trait, a trait that is determined by multiple genes where each dominant allele contributes a little bit to the overall trait value. What are some examples of traits that follow this pattern? You guys have no examples? Are you guys awake? I'm not, actually, I don't know what the genetic determination is of hair color. Everybody get out of your seat. Everybody up, move around. I want you to distribute yourselves such that the tallest person is up here, the shortest person is at the back corner. Go around the edge here. Order yourselves from tallest to shortest. Tallest up here, shortest back there.
Yeah. Wait, which back corner? Meg, I think this is back corner.
You don't have to be all the way back in the corner. You have to compare with other people to figure this out.
I just know I'm the shortest one in the class, so.
You were slouching, so it's like.
Yeah, I think you're taller. Yeah, that's good. Okay. What do you notice about this distribution? Okay, the first thing you notice is all the dudes are on one end and all the women are on the other end. What else do you notice about it? Is there just tall people and then short people? No. There's tall people, and then slightly less tall, then slightly less, da 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 da, Carly. There's continuous variation. There aren't these big steps. It isn't an either or thing. You're not garden peas. You don't have some tall garden peas and some short garden peas. You do have tall and short garden peas. You don't have tall humans and short humans like you have garden peas. There's this continuous variation. That continuous variation is because height is one of these traits that is determined by a bunch of different genes contributing to height. Each gene contributes a little bit to your height. Sit down. If we went out onto the campus and gathered data on all of the people on campus, measured their height and plotted a frequency histogram of it, what would that frequency histogram look like? It would look like a normal curve. Might be a little bit broad because men are generally taller than women. That's part of the sexual dimorphism in human beings. Dudes are just bigger than women because of hormones. So these normal curves usually indicate that there is something going on underlying that trait where it's governed by multiple genes, each gene adding a little bit to the phenotype, which is why we sometimes call these traits additive genetic traits, is because of this additive nature of them. We also use the term polygenic traits, although I will hardly ever use that term. We also refer to these as quantitative traits. Because the way in which we study these traits isn't by doing gigantic Punnett squares. What if you have a gene that is underlain by 20 different loci, or a trait that is underlain by 20 different loci? Genes at 20 different loci. You're going to be doing Punnett squares to figure that out? No, it's hard enough doing it with 3 traits, much less 20 traits. So the way we analyze these data is we use methods that are designed for analyzing distributions. What is the body of study that analyzes distributions? Statistics. We use statistics for this. Why do we start teaching about statistics early on? First and foremost, as we use it in this class, you're going to gather data. You're going to need to analyze that data. But then, once you get out of this class, you have to use it for another one, and then another one, and then another one. It's important. In this case, it's important for analyzing quantitative traits. So quantitative traits are these traits that are determined by multiple genes, each with a dominant allele, adding to the additive effect of genes on the phenotype. We study quantitative genetics. It's rooted in Mendelian genetics. I don't want you to lose sight of the fact that just because we're going to be analyzing things using statistical methods, that it isn't a set of traits that is governed by Mendelian genetics. It's governed by the same rules that pea plant height is governed by. It's governed by the same rules that pea pod shape is, and wrinkled versus round, and yellow versus green, and all that. Those same principles are what underlie these traits. It's just that you have to apply those rules on much larger scales because you have lots of genes contributing to the trait. So one of the ways that we look at quantitative traits, these additive genetic traits, is when we think about evolution, we can think of evolution as changing the shape and location of this distribution. So this is a trait distribution. Let's say that this is height. If there's selection acting on this trait distribution, we can see how that selection acting on the trait distribution changes this distribution of the trait over time. But what it's really doing is it's just changing what the frequency of the different alleles are in each of these different genes. So if we want to think about evolution, we can back up and go back to simpler versions of things. So we can go back to a single trait case. So here we have the most basic genetic cross, right? Who can work this genetic cross for us? Yeah.
I'm sorry, I'm a mess.
Do you need a Punnett square that big?
No, not really,
Not really, why not?
because there's only maybe 2 letters in each.
Each individual can only make one type of gamete, right? So you're going to make a bunch of offspring that are all the same gene type. So every individual is a heterozygote. Now we're going to take those heterozygotes and we're going to cross them, call on somebody to do the next cross.
From my group or a different group?
I don't care. Oh, I see what you're doing. You're trying to get points, aren't you? Dr. Judd, give the point hogs a point.
See you there.
Here at the back, yeah. They're hogging points because they need points.
Yeah.
This
This is true.
But he just gave you a point incorrectly because what should you be doing with Elizabeth? You should be helping her to make sure that what she's doing is correct.
Because it was a lot, it was a lot of.
So,
Big A, little
Cross
A.
big A, little a, big A,
Separate
little
out
a.
of this. There you go. Little A. Yep.
yeah, you're doing this.
You got it.
Okay, yep. Yeah, you're at this stage. Sorry, I wasn't keeping up with the PowerPoint. Okay, now your team has earned its point. So we have what Dr. Judd has already taken you through. We have a 1 to 2 to 1 genotypic ratio, and we have a 3 to 1 phenotypic ratio of whatever the dominant trait is versus whatever the recessive trait is, right? This is what we have out of this, right? How many alleles do we have in this system? We have 2 alleles. We have the dominant allele and we have the recessive allele. What is the frequency of the dominant allele and what is the frequency of the recessive allele when you just had these 2 individuals that you were crossing? How many alleles total do you have?
Oh, 2.
You have 4 alleles total. How many of them are the big A allele? 2. How many of them are the little a allele? 2. So in this first generation, the frequency of the big A allele is 1/2 and the frequency of the little a allele is 1/2. That was in the parental generation. The parental generation produced an F1 generation. It's made up of nothing but heterozygotes. What's the allele frequency here? You have 2 alleles.
Cool.
One of them is the big A.
1.5.
One of them is the little a. 1/2 in the F1. So this is the parental generation. This is the first filial generation. In the F2 generation, which is this, what is the allele frequency? You have 1, 2, 3, 4, 5, 6, 7, 8. You have 8 alleles total. How many of them are the dominant allele? How many of them are the recessive allele? 4/8 is 1/2. We could do this all day long. It gets a little more complicated because if we're going to cross these, we have to cross these with themselves at the frequency that they're at. We have to cross these with these, considering that they're at different frequencies. We have to cross these at the frequency that they're at. We have to cross these with these, considering their frequencies. Then we have to cross these with these at their frequencies. That becomes really complicated, but there's an easier way of doing this. We can replace the allele frequencies with P, representing the frequency of the dominant allele, and Q representing the frequency of the recessive allele, and basically put those allele frequencies in place. In this case, what's the allele frequency of the dominant allele? 1/2. What's the frequency of the recessive allele? 1/2. We can figure out then what the frequency of the homozygous dominant will be by taking 1/2, 1/2, multiplying that together to get 1/4. What's the frequency of the homozygous dominant? 1/4. Same thing, we have the frequency of the dominant allele times the frequency of the recessive allele. This represents 1/4 of the Punnett square, this represents 1/4 of the Punnett square, and then this represents 1/4 of the Punnett square. So the homozygous dominants will be represented by P squared, the homozygous recessives will be represented by Q squared, and the heterozygotes will be PQ times 2 because they appear in 2 different places. Does anybody in the room know what this equation is called?
Yes. Can I say it?
You can say it.
Hardy-Weinberg.
This is the Hardy-Weinberg equilibrium equation. It's an equation that relates allele frequencies to genotype frequencies. Let me say that again so that it's clear and you remember it. Actually, I'll write it on the board so it's clear and you remember it. What is P? An allele frequency or a genotype frequency?
Genotype frequency. Genotype.
What did you say, Carly? You said genotype. Who's right? What is P? Allele or genotype frequency?
Specifically the dominant allele.
It's the frequency of the dominant allele. How do we get genotype frequency out of that? We square it for the dominant allele. We multiply it times Q and 2 for the heterozygote. So these are genotype frequencies. If I tell you that the frequency of homozygous dominance is 0.49 in the population, makes up 49% of the population, what is the allele frequency?
Square root of 0.49.
Square root of 0.49, which is?
Uh,
0.7. That's the frequency of the dominant allele. Because this is an allele frequency, we square that to get the genotype frequency for homozygous dominance. If we know what P is, we also know what Q is. Because Q is whatever
1 minus.
P isn't. It's 1 minus P. So in this case it would be 0.3. What's the frequency of homozygous recessives in this population? What's that? Good.
3 squared.
Which is
0.9.
not 0.9,
Oh.
0.09. So if we know that this is the frequency of the homozygous dominance and this is the frequency of the homozygous recessives, that's 0.49 plus 0.09, that's 0.58. Then what must be the frequency of the heterozygotes?
2 times that.
1 minus 0.58, which will be 0.42. What's another way we can figure it out? 2 times P times Q, 2 times 0.7 times 0.3. What's 0.7 times 0.3? 0.21 times 2 is 0.42. P and Q are allele frequencies. P squared, 2PQ, and Q squared are genotype frequencies. If I give you an allele frequency, can you figure out genotype frequencies? If I give you genotype frequencies, can you figure out allele frequencies? In all cases?
I think so.
What if I tell you that the frequency of the heterozygotes is 0.49? You don't know the value of P or Q, do you? So, if I'm going to give you something like this on an exam, what do I have to give you?
P and Q. P or Q.
I have to give you either P or Q, or I have to give you
PQ.
P squared or
Oh.
Q squared. I can't just give you that because I could arrive at this a number of different ways by altering the value of P and Q. It'd be unfair. It's impossible to work out this way. You need to have a way of figuring out what P and Q are, and you can do that through the homozygotes because you just take the square root of the homozygous frequency. Everybody following how this works?
So what was the point 42 and what was that?
0.42 is the frequency of the heterozygotes. So remember, this is homozygous dominance, this is the frequency of heterozygotes, this is the frequency of homozygous recessives. Everybody jamming on a little bit of Hardy-Weinberg equilibrium here. What happened to allele frequencies from generation to generation as we went through this simple cross? Did they alter or did they stay the same? The allele frequencies stayed the same. Genotype frequencies varied, right? Because you went from no heterozygotes to nothing but heterozygotes, and then you went from nothing but heterozygotes to some homozygous dominants, some homozygous recessives, and some heterozygotes. But in all of that genotypic frequency change, the allele frequency didn't change. The reason that this is called Hardy-Weinberg equilibrium what do we mean in ecology and evolution when we talk about a system being at equilibrium?
Means nothing's changing.
It means nothing is changing. And in this case, the nothing that's changing, the nothing that's not changing, is allele frequencies. Allele frequencies aren't changing from generation to generation. So we could go through this equation generation after generation after generation after generation, use the previous generation's allele frequency to tell us what the next generation's genotype frequencies are going to be. And guess what? It's going to be the dullest exercise on the planet because it's always going to be 1/2 big A and 1/2 little a. Because that's what we started with because this system isn't evolving. Which brings us to Manx cats. Has anybody ever seen a Manx cat? Dr. Judd and I have seen a Manx cat. I just saw a Manx cat recently, actually, in my neighborhood. So the cat lover in the class and 4 others have seen Manx cats. Which tells you what about Manx cats?
Very common.
They're probably not very common. So one of the questions as a biologist that you can ask is, why are Manx cats not very common? Well, Manx cats are not very common because Manx catism is determined by a dominant lethal gene. A dominant lethal means that it's lethal, but it's only lethal in the in the homozygous dominant condition. So homozygous dominant individuals are spontaneously aborted before they are born. They're called Manx cats because they were first reported on the Isle of Man. And so you can't have a homozygous dominant cat. A homozygous dominant cat is dead before it ever is even born. So at best, you could maybe find one of the Manx cats that Carly found and one of the Manx cats that Luke found and get those Manx cats together because you're going to breed a population of Manx cats because you think they're cool looking. Well, you know why. What's the allele frequency in this population? Frequency of the big M is 1/2. I'm actually going to do it with fractions. I find that easier. The recessive allele is 1/2. This is P. This is Q. Somebody not in their group come up and cross these 2 Manx cats.
This is a Russia.
First step is what? You guys always want to jump to the Punnett square. What should you always stop and do first? Who? What's that? Who? Oh, who's? I thought I saw your lips move.
Is the, you want to find the genomes, don't you, or whatever it's called?
You want to first make gametes.
Yeah.
What kind of gametes can each of those individuals make? Are those gametes? Those are not gametes. Those are genotypes. What kind of gametes can they make? gametes are always haploid, you're starting with two diploid parents, right?
You said what?
What type of gametes can they make? So these guys have two copies of each gene. These guys have two copies of each gene. The gametes have to be haploid because when you combine them together, you want to get a diploid offspring. So what gametes can this individual make? One will be a big M gamete, one will be a little M gamete. Now you can proceed to making a Punnett square. The reason I have you make gametes first is because when we're dealing with these standard kinds of crosses, figuring out the Punnett square is easy. But if I give you a non-standard cross, figuring out gametes won't be as obvious. And so you have to think about gametes more sometimes. Okay. What do you know about the phenotypes of these four individuals?
Homozygous dominant, yeah.
The homozygous dominant is dead. We have two
Heterozygous.
heterozygotes, which will present as
Alive.
alive but
Carriers.
Manx cats. They have the short
Oh, yeah,
bodies and the
Manx.
square tails. And then what is the homozygous recessive?
Normal cat.
Normal cat. So if we don't think about these dying, if we think about at conception we have a bunch of little baby kitten zygotes, single cells. The allele frequency here is we have 8 alleles. 1, 2, 3, 4 of them are dominant, and 1, 2, 3, 4 of them are recessive. But because you don't make it from zygote to born cat, if you're this, you don't have 4 parts of your population anymore. You have 3 parts of your population. And of those 3 parts, 2 of those are heterozygous, and 1 of those is homozygous recessive. 2 Manx cats and 1 normal cat. Now how many alleles do you have?
6.
You have 6 alleles. How many of those alleles are the big M allele? 2 6, or 1/3, is the big M allele, and 4 6, or 2/3, is the recessive allele. So our allele frequency has changed. Looks like this. Allele frequencies were 0.5 and 0.5. We get rid of the dead cats, and it goes to 0.33 and 0.67, which is just the decimal equivalent of 1/3 and 2/3. Now, if we're gonna cross these guys again, we have to do what? We have to cross all possible combinations of these relative to the relative frequency of them in the population. But that's a pain in the butt to do, so what can we just do? We can use the Hardy-Weinberg equation. How can we use the Hardy-Weinberg equation?
Plug and chug?
Hmm?
So plug and chug? Because we know the numbers, right?
Do it. Oh, I just threw it at you anymore.
Yeah. We've lost so many opportunities in getting points.
Oh yeah, so who was answering before? Oh, Bronson.
Yeah.
Bronson's team gets a point. I like days like this because points fly. So do markers, but sometimes it doesn't go very well. Can you do it with fractions? It's going to be actually easier with fractions. And I'll show you why in a few minutes. Yeah, chug away. You said plug and chug.
I don't know the calculator.
You don't need a calculator. What's the square of 3? 1/3 squared is no 1/9, because you have to do numerator and denominator, not 0.9.
What?
Fraction.
Oh, sorry.
You're just doing 1/3 times 1/3. 1/9. There you go. I'm making her do fractions because I'm going to show you something in a minute that will say, oh, that's easier to do with fractions than it is to do with decimals. That is right. 2/3 times 2/3 is 2 times 2 is 4, 3 times 3 is 9. That should all equal up to 1, which is
That's right.
9 ninths, right? 4, 8, 9 over 9 to 9 ninths. Meg, point for your team. She plugged, she chugged, she prevailed. But of course, what is this 4 ninths? Is it an Allen frequency or a genotype frequency? Genotype frequency of what genotype? Homozygous recessives. Genotype frequency of heterozygotes. Genotype frequency of homozygous dominants, which are also dead. And I'm down giving away all my markers. Up here. So those guys are dead. So do we have 9 parts of a population anymore? No, we have 8 parts of a population. So we only have really 8 eighths. This is why I wanted you to do fractions. So do we have 4 ninths and 4 ninths, or do we have 4 eighths and 4 eighths, don't we? So it basically 4 eighths reduces down to 1/2 and 1/2. So it's like having a population of only 2 cats, right? The genotype of 1 cat is, or heterozygous, as we would say in sciencey classes. The other half would be homozygous recessive. Good. How many alleles do we have? We have 4 alleles. How many of them are the dominant allele? 1/4. How many of them are the recessive allele? Matt, call on somebody not in your group to work through the next generation.
Riley.
Here.
While he's doing that, if you work through the PowerPoint, I basically just repeat what was just on the board in all of its glory. Because I'm not very creative.
Didn't you just give us a lecture the other day about how you have to be creative?
What's that?
Didn't you just give us a lecture about how you have to be creative to be scientists?
Yeah, but then scientists aren't very creative in some cases. What do we call it? Hardy-Weinberg equilibrium. Dude named Hardy and a dude named Weinberg came up with it. Not very creative. Why do we call it Punnett square? Dude named Punnett, Reginald Punnett, came up with it. Not very creative. So in some ways you should be creative, and in other ways we're just dull. What do you got? What have you got there? Okay, so what is that? 6/16 and 9/16? That's actually. Oh yeah. Okay. All right. Gotcha. That's so. Yep. Gotcha. So, walk us through it. Those guys die. So do you have 16 parts of the population, or do you have something less? You have 15 parts of the population. 6/15 is the,
That's the heterozygous.
and 9/15 is the homozygous recessive. 6/15 and 9/15 breaks down to something easier. If you divide both by 3, the numerator of one becomes 2/5, the other one becomes 3/5. What's happening to allele frequencies? They're changing. In what way, specifically? What's getting smaller? Is 3/5 bigger than a half or smaller than a
It's
half?
going like this.
It's bigger than a half, right?
It's going like this.
Right?
It's a normal distribution.
No normal distribution here. No.
Damn.
This one's getting smaller and smaller, less frequent and less frequent. This one is getting more and more frequent. If we did this another generation, what would it be?
Smaller than that, so.
It would be 2/5 squared plus 2 times 3/5 times 2/5 plus 3/5 squared. So this would be 4 25ths plus, wait, is that right? Plus 6, 12, 25ths plus, this doesn't feel right.
We didn't multiply it, but.
Yeah,
So,
Yeah,
it's supposed to
we
be
didn't
10, right?
Oh.
multiply
It's
that.
10.
Yeah.
No, that's right, that's right. Yeah, that's right. No, that's right. 4,
why am I confused?
I don't know, it's huge.
The two PQ seems wrong to me.
Oh, then you drop the 24, that becomes 1/2, and that becomes, it's 9 and 24. Oh, it's not even, doesn't go, doesn't work.
Yeah, so that's 3/8.
That's 3/8? So it's 1/2 and 3/8, is that?
Oh, wait.
1/2 and 3/8?
No, it's because we did something wrong in the last step.
Oh, I thought it was alright.
Yeah, we were rushing through things.
Oh,
We
no,
were
it's
rushing
okay.
through things. So we had 1/4 squared plus 2 times 1/4 times 3/4 plus 3/4 squared. This was 1/16 plus 6/16 plus 9/16. That's 16/16, but these guys are dead, so that's actually 6/15 and 9/15, which reduces to 2/5 and 3/5. Are these genotype frequencies or are these allo frequencies?
Genotype frequency.
These are genotype frequencies. Genotype frequencies don't go down here. These are allo frequencies. So this is like saying we have 2 homozygous dominant, sorry, we don't have any homozygous dominants. We have 2 heterozygous individuals and we have 3 homozygous recessives. How many alleles total do we have? 2, 4, 6, 8, 10. We have 10. How many of those are the dominant allele?
2,
How many of those are the recessive allele?
8.
I knew it was 1/5 and 2/5, or 1/5 and 4/5. I don't know why I was continuing down your path of misleading me. So when I stressed earlier the difference between genotype frequencies and allo frequencies, if you don't make that distinction, what happens? Your math goes wonky, doesn't it? Your math goes wonky. So you have 2/5 and 3/5. These are genotype frequencies, not allo frequencies. Same thing that's on the board is on the screen. And what will happen eventually? Don't go to the next slide. This is where you don't go to the next slide. What's going to eventually happen to these allo frequencies? What's going to happen to P in particular?
It's gonna go away eventually. It's gonna disappear.
Okay, so some of you are saying that it's going to go way down, and some of you are going to say it's going to go way down until it's extinct. We normally think of alleles as going to extinct. But what would extinct mean for this? It goes down to a frequency of.
0.
0.
All right, who thinks it'll go to a frequency of 0 eventually? Raise your hands.
Just like,
Oh, like super close?
0.
like super close to
0.
like a zero.
No.
Just 0, 0.
No, not
It's
0.
gone, just totally gone. Gabby still thinks it's going to be totally gone. How many of you think that it's not going to be totally gone? Okay, why do all you guys think that you're right and Gabby's wrong?
You can't get to zero by dividing.
Hmm?
You can't get to zero. If you just keep going, it just keeps getting smaller. We'll just start to drop off.
So why?
There's gonna
But why?
There's always gonna be heterozygous progressive. Think about it.
It's going to always be harbored in heterozygotes. The dominant allele is always a little bit safe because you don't have strong enough selection to get rid of it. There's strong selection against the dominant allele because if you have the homozygous dominant condition, you die. But there's no consequence for having the heterozygous condition, so there's always a place for that dominant allele to hang out. Do it long enough, this is what it looks like. It gets close to 0, but you can't ever get rid of it totally. What would you have to do to get rid of the dominant allele altogether? You would have to go on a Manx cat killing spree, wouldn't you? Otherwise, you're always going to have a small frequency of Manx cats in the population, which means you're never going to get rid of that dominant allele. You have to engage in Manx cat genocide, which we're not going to do, probably. So it gets smaller and smaller and smaller, but it gets to the point where it's so low that it really decreases at a decreasing rate over time. So it's harbored by the heterozygotes, and in order to totally rid the Manx cat of the population of Manx cat genes, we would have to kill all of the Manx cats that we see. And so Manx cats are rare, which is why only a handful of us have seen them in our lives, but they're still out there because we don't do that to cats. The same thing happens in natural selection happening on quantitative traits. If we take that initial population that we started with, we cross it, we got this distribution where we have one of these guys, we have two of these guys, and we have one of these guys. But what happens to the one of these guys? They die. What does this initial distribution look kind of like? It looks kind of like the beginning of a normal distribution, just you have very few genes contributing to that normal distribution. And what did we get rid of? We got rid of one end of the distribution. That's one definition of selection. In this case, it's selection against homozygous dominance. When we go back to this situation, there's a lot more genotypes, so there's a wider distribution of the number of dominant alleles in the genotypes of the resulting offspring, but if there's strong selection against the dominant allele, you're selecting against this side of the distribution, not this side of the distribution, if the selection is against dominant alleles, the phenotypes that dominant alleles result in. If the selection is against the phenotypes that recessive alleles result in, then there's selection on this side of the distribution, and it pushes the phenotype up in value. If you have selection against dominant alleles, it pushes the distribution to the left. Hardy-Weinberg equilibrium basically says that from generation to generation, allele frequencies do not change unless what?
Someone's killed.
Pardon? Unless selection occurs. So Hardy-Weinberg equilibrium states that allele frequencies within a population will not change when selection is acting on a population. If selection acts on a population, it means that some alleles don't get inherited as much as others because they get removed from the population, because Manx cats that produce homozygous dominant individuals, those homozygous dominant individuals die before they are conceived. That's what selection is. When your survival or reproductive success is tied to your genotype or your phenotype. what other things can happen that we've already studied about in this class that can alter allele frequencies over time? Yeah. But that would be a type of selection. If your phenotype has something to do with you being able to survive predatory attachment. Yeah.
Immigration.
Yeah, so migration, moving into and out of populations. I got a postdoc after graduate school in Puerto Rico. You will notice I am white and I have blonde hair. When I have hair, it's blonde. It used to be blonde, now it's probably gray. I haven't seen it in a while. I was a white blonde-headed dude headed to